there's a protein just before the cell, how about this? There's a protein on XYZ that's involved in septation, breaking the cell into the $2 cell. Yet we're not going to find it in cells that are in B growth. We're not going to find it in those cells that are in C growth. It's only going to be those that are in late D growth. So we need to get as much of the population in late D growth as possible so that we've got a population cell we can work with, so we can purify that material. XYZ. Does that make sense? Maybe. Well, it is important that we do so, because this is how we get the workable quantities of these enzymes and proteins. The next question is, how would we synchronize the culture? How would we synchronize it? Any ideas? Let's take an example of something you would be familiar with. In the class we use the example of Kentucky Derby. Horse race. First of May, I think, somebody the first one Saturday. Everybody heard of the Kentucky Derby? Everybody seen the Kentucky Derby? Maybe not. But in any horse race, you suppose they just shoot all the horses onto the field and then run around and whoever comes around first is the winner? That's not the I saw a horse race at all. Yeah, in a starter gate, yes. In a starter gate. In other words, you bring all the horses, all the jockeys onto the field, and you put them into a starter gate. They're all brought into that gate, and they're left right there. At that point in time, they're all at the same point on the track. So how could we get cells physiologically at the same point of their metabolic metabolism? Don't take away all of them. Take away one. Take away a vitamin. Take a vitamin. All the cells then will metabolize until they get up to the point where they need that vitamin. Without it, they stop. They stop metabolism. They stop growth. And depending upon the culture, depending on how fast they are capable of growing, typically like 3 or 4 hours, you say, okay, all the cells should be at the point where they need vitamin XYZ. Now I'm going to put vitamin XYZ into it, and they're off to the races. Now the cells take XYZ, they utilize it, they go to the next step. The next step, the next step, and then they're dividing at the same rate. At that point, they're all at the same part of the cell cycle simultaneously. They're simultaneously at the same point of the cell cycle. Now, even in a clone of cells, some of them are going to start to do things faster, and it breaks down after about 3 of those cell divisions. But that's how we study enzymes and proteins that are found in very low concentrations and only at a particular point of the cell cycle. By lining them all up so that we can get them to the same point and then capturing them, purify this and isolate XYZ and study it. Okay. Let's go back to our closed system. Bear with me. If I don't add any more nutrients, if I don't neutralize toxic materials in that closed system, it will ultimately stop growing at that particular rate of defined by decay value, and it will go into the next phase that we call stationary growth. But are there instances where we might not want that to happen? Are there instances where we would want to keep the growth in exponential phase? Engineering. Let's suppose that this population of E. coli cells has a plasmid. Now we talked about our plasmid, we talked about your plasmid, we talked about the engineered plasmids that contain genes of interest, human insulin, for example. And you've got your bacterial cells here, they're rapidly dividing, they're metabolizing very quickly, they are transcribing and translating that human insulin gene, and those bacterial cells are producing lots of human insulin. And as long as we keep them in log exponential phase growth, they're going to produce lots of it. So do we want that culture to go, to stop? No. No, we want to keep it there. How can we keep it there? Well, we can't keep it there in a closed system. We have to use what's called a chemostat. Chemostat. Another term, which is beginning books will use the term interchangeably. There is a difference. There's what's called a turbidostat. Chemostat or turbidostat. Turbidostat measures turbidity. Obvious in the and here you can see that this is not a closed system because we've got a carboy, a fresh medium here. We can control the amount going into our culture vessel. This is our culture vessel. You can see that we've got air going in here. Oftentimes there'll be a little stir in there that certainly keep the cells up off the bottom. But notice this level right here. This level is such that it is just level with this. And we drop one drop of medium in here, it overflows and one drop of medium drops into the receptor. It's called catch basin. All right, I drop 10 drops in, then 10 drops fall out. These are cells that are producing my insulin. This is supernatant that they have the insulin in it. So all I have to do is just add fresh medium at a periodic rate, and I can keep that culture in while the working growth. Chemostat or turbidostat allows for continuous exponential growth. Yeah. From an industrial standpoint, it allows for the collection of product. If these cells are producing insulin, then we can capture the insulin in the supernatant. Somebody takes that little bucket over to the lab and somebody begins to purify it. And that little bucket you collect sometimes it can be very large, it can be hundreds of gallons every hour, depending upon the vessel. Now you say, well, look at this. What in the world? How did you get 100 gallons? Well, in industrial circles, a chemostat is oftentimes referred to as a fermenter. The fermenters can be 200,000 to 300,000 gallons of liquid medium. They can be 2 and 3 stories high. Lots of medium, lots of bacteria growing in it, lots of product being produced. From an academic standpoint, it allows for a detailed, continuous analysis, kinetic analysis of bacterial growth. Much of what we know about bacterial growth was actually done in a chemostat. How much of what we know from the chemostat is found in the real world out there in nature? There's some people who say, well, it's probably more artificial than what is really real. This is, you don't have this picture, but this is an example of laboratory chemostats. Each one of these little things here are ports that you can put in a temperature probe, you can put in some type of stirrer if there need be. We've got ways here that you can monitor the oxygen concentration, all sorts of parameters on the road. And on a fermenter, on each story, you're all the way up to the length, the height that you're going to have gauges telling you exactly what the dissolved oxygen is, what the pH is, what the concentration is, all sorts of things. Okay. So, let's come back. Let's come back to this. Let's suppose that you work for Eli Lilly. And some of you may work for Eli Lilly as retail pharmacists. I know a lot of pharmacists who ended up at Lilly. Let's suppose that you do work for Eli Lilly and you're in charge of antibiotics. Antibiotics. Well, we've said that antibiotics are going to be produced right here. How can we adjust our chemostat so that we maintain that culture there and not down here where we're getting primary metabolism? You know, what primary metabolism? by adjusting the flow rate, I can move that population from the metabolic standpoint. I can move them up, or I can move it down. I can determine by adjusting that flow rate, I can move that population so they're down here in the salad strip. Or I can typically slow it down and then move up here to the secondary. So if I want antibiotics, I'm going to have a relatively slow flow rate into that hemostat. Now, let's give you some rates. How about 10 liters per minute? And then we can go all the way up to 200 liters per minute. That's a lot of volume, but we're not talking about that little tube depicted here. At 10 liters per minute, am I going to be maintaining this population here in the secondary or the primary? What is it? One minute. Slower rate. Imagine this, take it to zero. If I'm not having any fresh medium, what's going to happen to this culture? It's going to come up here and go beyond the secondary and into that stationary. So I need to be adding a certain amount of medium to keep it down here. It's been determined, I can slow that rate down, maybe it takes 15 liters per minute to maintain it right there. But if we decide, and we wouldn't do it in the same culture, we wouldn't do it the same per minute, we want to say, okay, well, we're going to switch now to these E. coli to producing citric acid for food production. Citric acid is produced lots of food manufacturing, food processing use citric acid. Lots of food industries will get tank cars with citric acid every day. And it's produced all biologically, all microbiological. But that's a primary metabolite. We want to shoot that down to here. And the way to do that is increase the flow rate. Now, I could increase the flow rate so high that I could actually drive the cells back into the lag phase. Because I would be diluting them to the point that all of the dividing cells have basically been removed. Now, it would very quickly come back because these cells that are find themselves down here are going to have just come out of cell division and they're in what we call dew phase. But they'll come out. But I can drive it down there, and all by adjusting that flow rate. That makes sense? And there's our chemostats, there's our vivistats. The next thing we're going to talk about is the microbiological assay. I'm purposely not giving you anything on it other than a sheet that says microbiology. We don't have time to get started with this, so we will stop at this point. We will pick it up on Tuesday. In preparation for this, how many of you have ever used a standard curve? In chemistry, sometimes they may have colorimetric. They'll have a standard curve, and then you measure the optical density, and from that standard curve, you can determine the concentration of an unknown. No idea what I'm talking about. And then you might make dilutions